We discussed fatty synthesis in the previous class. So what were the three steps? First was we needed raw materials, that was acetyl CoA and NADPH, right? And one was the ATP. Yeah, right. The second step was conversion of acetyl CoA into malonyl, right? And third, we did fatty acid synthetase complex, right? Right. These was discussed in previous class. Today we discuss reactions of fatty acid synthetase complex, right? Uh, can I revise it for 5 minutes? The previous class, okay? So what was acetyl CoA, what was NADPH, and what was ATP, right? They were raw materials for the synthesis of fatty acid. What was the function of this molecule ATP? It was supposed to provide energy, right? And this NADPH will provide reducing equivalents, right? And what were the sources of NADPH? One was HMP shunt, and the other name for HMP shunt is PPP pentose phosphate pathway. Second sources was isocitrate dehydrogenase. Third was malic enzyme, right? The sources were reducing equivalents are done. ATP is done. Now one is acetyl CoA, right? What is the source of acetyl CoA? Acetyl CoA sources. First was we discussed glycolysis, right? We start from glucose, glucose 6 phosphate, fructose 6 phosphate, fructose 1 6 phosphate, and finally we form pyruvate, right? So pyruvate becomes the first source. How? Pyruvate is converted via pyruvate dehydrogenase reaction to form acetyl CoA. Done. The first source of acetyl CoA. The second source is beta-oxidation, right? In beta-oxidation of even chain fatty acids, right? We discussed the end product was at the fourth step, it was acetyl CoA and acyl CoA, right? So in the fourth step, we also got acetyl CoA, right? The first source was pyruvate from pyruvate dehydrogenase reaction. Second is beta-oxy. Third is ketogenic amino acids, right? Clear? So we did all the three sources. First was NADPH, second was ATP that provides energy, NADPH provides reducing equivalents, and NADPH is got from HMP shunt or pentose phosphate pathway, and the second source was isocitrate dehydrogenase reaction, and the third was malic enzyme reaction, right? And the first source or raw material is acetyl CoA. It is got from three sources. One is pyruvate dehydrogenase reaction to form acetyl CoA. Second is beta oxidation of fatty acid, even chain will get acetyl CoA. Third is ketogenic amino acid, right? Now we have all the raw materials. That is our first is acetyl CoA now, right? But it is present where? The problem was that it is present in the mitochondrial matrix, right? And this fatty acid synthesis takes place where? In the cytosol fraction of the cell, right? So what we will do? We will bring it from matrix to the cytosol, right? So that we can synthesize fatty acids, right? What will we do? We do compartment reactions like this. I draw the diagram. So this compartment is our mitochondria or matrix, and this is our cytosol, right? So first of all, we undergo glycolysis from glucose. What will get? Pyruvate, right? Pyruvate is there, that is will be permeable into the matrix, right? So pyruvate will come inside into the matrix, right? This pyruvate via pyruvate dehydrogenase reaction will form acetyl CoA, right? And it can also form oxaloacetate via carboxylase reaction, right? Now we have these two. These two will combine with each other in the presence of citrate synthase, right? What will get? Citrate. right? The problem is that this acetyl CoA formed is not permeable from matrix into the cytosol, where we will synthesize fatty acids, right? So this citrate is there from acetyl CoA and oxaloacetate reaction, we get citrate. Citrate will be permeable outside. The citrate comes out. What is this compartment? This is our cytosol, right? The citrate comes outside. Now this was citrate synthesis because we are synthesizing citrates, right? This is citrate lyase enzyme because we are breaking down. So it will get oxaloacetate. Oxaloacetate will convert it into malic, by a malic enzyme it will form malic, and then what is formed? Pyruvate. Now pyruvate formed is regenerated by this reaction as well, right? And in this reaction we also get our the reducing equivalent, that is NADPH, right? Now the three raw materials was, one is acetyl CoA. Now it is here in the cytosol, right? The three raw materials was acetyl CoA, then NADPH, second, and third is ATP. Now all the three raw materials are there in the cytosol. Now we will start with fatty acid synthesis. For fatty acid synthesis we require fatty acid synthetase complex, right? And I told that you can get 5 marks question for the diagram of fatty acid synthetase complex. You have to write the enzymes. It can also come in multiple choice reactions, right? So let's revise it as well. So what was the mnemonic? First was KS, second was at, third was Mother Teresa, fourth was Delhi, fifth was Keto, no sorry, Eno. For relief we got Eno, right? Eno relief. Sixth was Keto. Then Keto was caught by ACP of police, and this is attached to this, and the seventh enzyme is thioesterase, right? KS Kabir Singh at Mother Teresa Hospital, New Delhi. Why? Because he was suffering from acidity, right? After acidity, the doctor, he went to Mother Teresa Hospital. The doctor advised him Eno, right? So he got relief from acidity. So he was okay. In the morning, he went to the pharmacy shop and asked for more Eno packets, right? But when the pharmacy went checked his stock, he found that the Enos were stolen. So he checked the CCTV footage, and in the CCTV footage he found that there were lots of ketos residing there, ketos or kitties. They stole that Eno packets, right? After the packets were lost, the police was called, and ACP caught that keto, and he handed over to the thane or the police station. The enzyme is thioesterase, right? Kabir Singh is our keto acyl synthetase. At is our acetyl transacylase. Empty Mother Teresa is malonyl transacylase, right? Delhi for dehydrogenase. In some books it is hydratase written, in some books it is dehydrogenase written, right? Then Eno for enol reductase, right? Keto for keto acyl reductase. ACP is acyl carrier protein, and thioesterase for thane, right? Thioesterase, right? This is one monomer. The other monomer, the dimer is functionally active, I told, right? The other will come like this in the diagonally, right? So this will come here, KS, AT, MD, and Delhi, then Eno reductase, then keto reductase, then ACP is attached, then our thioesterase, Clear? This is very important. Your mnemonic will help you learn and you will get 5 marks. No problem, right? Now coming on to today's class, what is topic? The reactions of fatty acid synthetase complex, right? All the raw materials are in the cell now. In the cytosolic fraction, we have acetyl CoA inside, we have NADPH inside, and we have ATP inside. We have energy and we have fatty acid synthetase complex. Now we synthesize fatty acids, right? The first reaction was conversion of acetyl CoA into malonyl-CoA, right? When acetyl CoA is brought into the cytosol, it is converted into malonyl-CoA, right? And this reaction requires the mnemonic is ABC. A, B, C. A for ATP, B for biotin, C for carbon dioxide. As simple, right? So acetyl CoA is converted to malonyl-CoA via A, B, C. A for ATP, B for biotin, C for carbon dioxide. And the enzyme is acetyl CoA carboxylase. Carboxylase why? Because we are adding carbon dioxide. Clear? Now we have malonyl-CoA. In the cytosol, the reaction is going on for fatty acid synthesis, and this acetyl CoA is converted into malonyl-CoA, right? Now what will we do? Suppose we have acetyl CoA and malonyl-CoA. Now I am starting with the reactions of fatty acid synthetase complex, right? So first we will have acetyl CoA and second is malonyl-CoA. Remember one thing I checked yesterday, the reactions are different in Harper, bit different in Vasu and bit different in Sutherland, right? So I picked from Harper, right? So you can cross check them, but I think Sutherland has some bit mistakes, right? You can get this video as well, so you can do it from this side, okay? So we have acetyl CoA and malonyl-CoA, right? What we use? We use acetyl transferase and malonyl transferase. We will get acetyl ACP. Acyl carrier protein is now attached, one of the part of, right? And here we will get malonyl ACP, right? Now these two will undergo condensation reaction. Condensation means they will condense. This is 2 carbon, this is 2 carbon, but we will get acetyl ACP, right? Please have a screenshot of this, or you can write it on your notebook, right? After that, now we have acetyl CoA, acetyl ACP. This is 2 carbon, this is 2 carbon, this is 4 carbon, right? And from acetyl CoA we have reduction reaction, right? Reduction is going on, and for reduction we use the reducing equivalent, name the reducing equivalent NADPH. We got it from HMP shunt, right? So we'll add NADPH and it is converted to NADP, right? What will get? कोई पढ़ के आया अपने आप पहले बोलो? We get hydroxy acyl ACP, right? In the next section, we do dehydratase. Dehydratase means we are removing water. So water will come out. What will get? Anybody? Tell me. After hydroxyacyl ACP, you will get protonyl ACP, right? After protonyl ACP, again reduction will come, second reduction, right? For reduction, the same reducing equivalent NADPH, it is converted to NADP positive, and what will get? Butyryl, BUTYRLAC. Butyryl, how many carbon atoms? Methane, ethane, propane, butane. 4 carbons are there, right? So we'll get 4 carbons. Please get screenshot of this. So now we have butyryl. Butyryl means 4 carbon, methane, ethane, propane, butane. Butane contains 4 carbon. So we have 4 carbon acyl carrier protein, that is butyryl ACP, right? In the previous class, I told you what is the function of acetyl CoA. It is the source of carbon atom, right? So acetyl CoA is source of carbons. And how many carbons does it contain? 2 carbons, right? This is the first step. Up to 4 actions will form. The first thing is that will form 4 carbon fatty synthesis complex with butyryl ACP. After that, the again 1, 2, all the 4 actions are repeated, right? Until you get the 16 carbon palmitic acid. How? We have 4 carbon plus 2 carbon via acetyl CoA, right? Again the steps are repeated. Then 6 carbon plus 2 carbon via acetyl CoA. The step is repeated up to approximately 7 to 8 times, and finally we will get our 16 carbon palmitic acid. And it doesn't contain double bond. How will you add double bond? How will you add double bond? Any guesses? Backbenchers. Vivisha, you are sitting at the back. Why? Why a backbencher? You are not supposed to sleep in class this time. Come here. Yeah. Come here. Yeah. We are getting finally the 16 carbon palmitic acid. 16 carbon palmitic acid. Right? And what are the essential fatty acids? Tell me. Essential fatty acids are mostly PUFAS, polyunsaturated fatty acid. Tell me the names. Linoleic, linolenic, and ketogenic acid. We are forming the 16 carbon now only, but these all contain double bonds. Now how will you introduce double bond? This is a saturated, right? And these are unsaturated. So we have to introduce double bonds, right? But in humans, you cannot introduce double bonds beyond carbon 10, right? The body has the capacity to introduce double bond up to carbon 10. After that, body cannot introduce double bonds. What will the body do? Body surrendering to the conditions. So you have to eat them. That is why they are essential fatty acid. You have to take them into the diet, right? Tell me sources of good omega-3. Just raise your hands. Omega-3 good sources for your good brain functioning, like me. Tell me. फिश तो तुम खाते ही बहुत हो इधर, क्या करें? वेजिटेरियन के लिए बोलो, मेरे जैसे बंदे के लिए बोलो। Any guesses? Best is flax seeds. Flax seeds. I don't know what you call it in Bengali. We call it alsi in Punjabi. Alsi, flax, right? Any other sources? Chia seeds. Chia seeds. Any other? Any other? She's laughing. She's enjoying, right? The two main sources are flax seeds and chia seeds for omega-3. If you are eating omega-3 in proper manner, what will happen? You will not have depression, you will not have anxiety, the brain will function properly, neurons will fire properly, right? You will not gain weight. The girls who have PCOD, that will not appear, right? Fatty liver will also sooner or later it gets okay. Lungs problem will not come, no asthma will come if you are eating omega-3 in right manner, right? And sources of fish, good fish, omega-3, fatty fish. The fish that is rich in fat is good, omega-3. और ये अगर ये फिश यमुना के पानी की है तो मत खाना। जो टॉक्सिक फिश के अंदर है वो आपकी बॉडी के अंदर जाएंगे, राइट? फ्रॉम गुड सोर्स, गुड वाटर सोर्स, राइट? उसमें केमिकल ना हो। अगर यमुना का पानी यूज करोगे उसकी मच्छी खाओगे तो गड़बड़ हो जाएगी। चलिए, राइट? एंड अब सो वी जस्ट रिकॉल वी हैव थ्री सोर्सेज, एसिटाइल कोए and NADPH and ATP. The first process was acetyl CoA was introduced into the cytosol from mitochondria, right? In the second step, we converted acetyl CoA into malonyl-CoA, right? After that, we use the four reactions in which acetyl CoA and malonyl-CoA was converted into by transfer reaction acetyl ACP and malonyl ACP, right? Dial ACP plus malonyl ACP, right? This is 2 carbon, this is 2 carbon, right? We did condensation reaction. After condensation, what will get? acetyl ACP. This is again 4 carbon, 2 carbon, 2 carbon is equal to 4 carbon, right? Then the first reaction is reduction. Then the second reaction here is first. The second is we are moving water, that is dehydrogenation reaction, right? Then in the third step again reduction. What will get after reduction? Butyryl ACP from crotonyl ACP, right? That is butyryl. Butyryl it contains 4 carbons, right? Then again the steps are repeated by adding one molecule of acetyl CoA, right? Acetyl CoA contains 2 carbons. So we have 4 carbon fatty acid, 4 plus 2 is equal to 6 carbon, right? Again repeated the whole process, we will get 8 carbon, then 10 carbon, 12 carbon, 14 carbon, and finally 16 carbon. That is our palmitic acid. Clear? And acetyl CoA ACP was formed from acetyl CoA, and this was formed from malonyl CoA. This is acetyl transferase, and this is malonyl transferase. Clear? So how will you remember the mnemonic? Just try to recall. We have 2 reduction reactions. When we will form ACP, both from the beginner's product, that is acetyl CoA, so we have 2 carbon, 2 carbon, we will get acetoacetyl ACP, that is our 4 carbon, right? So in this reaction, this is condensation. We are condensing 2 carbon and 2 carbon will forming 4 carbon, right? After that, the mnemonic is 1, 2, 3 reduction, then dehydration, and then again reduction, right? And for reduction you are using reducing equivalent NADPH. So both reactions will require NADPH, and this will also require NADPH. Like this NADPH and this NADPH, and one is dehydration. In the second step, you are removing water. Clear? So it is done now. We are finally formed via to adding two carbons. We have finally formed 16 carbon. That is our palmitic acid. Now problem is coming about double bonds and unsaturation. We'll discuss now. This enzyme will cause desaturation, right? Desaturation कहाँ से होगी? Desaturation means you are adding double bonds. So it is added by enzyme fatty acyl CoA desaturase. This is the enzyme, right? And it is what type of enzyme? What does it require? It requires cytochrome B5, right? NADH. Anything else? Anything else? Tell me. And molecular oxygen as well. First step is introduction of acetyl CoA into the cytosol from matrix, right? You have acetyl CoA inside the cytosol, you will convert it into malonyl-CoA, right? And this malonyl-CoA is precursor for further fatty acid synthesis via fatty acid synthetase complex with 7 enzymes. We discussed it. In the first step, acetyl CoA and malonyl-CoA converted into acetyl ACT and malonyl ACT via transferase reaction. Then those 2 carbon and 2 carbon will condense to form acetoacetyl ACT. Then via reduction protonyl CoA, again dehydration butyryl CoA, and finally we'll get 4 carbon butyryl, right? And here we got 4 carbon. After that you have to add 2 carbons by acetyl CoA via the same process. So 6 plus 2, 8 plus 2, 10 plus 2, 12, 14, and finally 16 carbon palmitic acid is formed, right? Now, to introduce double bonds, you need this enzyme fatty acyl CoA desaturase. Desaturation means it introduces double bond, right? Unsaturation done हो रही है. Then it requires cytochrome B5, NADH, and molecular oxygen, right? So what will you get from palmitic acid? We found palmitic acid here. It is saturated or unsaturated. It doesn't contain double bonds, means it is saturated. And after you use this enzyme, it is converted into palmitic is there, and it is converted into palmitoyl-CoA because we introduce double bond via this enzyme. Right? Now we have introduced double bonds, or suppose you have oleic to palmito-oleic, right? And I told the PUFAS, this PUFAS, that doesn't contain more double bonds beyond carbon 10. EUFAS. So humans lack to reduce double bond between carbon 10, right? So we have to take this into the diet, right? That is why they are essential fatty acids, because the body cannot synthesize them. And one more thing, normally the body can form up to 16 carbon, but we have arachidonic acid. How many carbon atoms are there? 20. So how will you further elongate the chain? Any 18 carbon you discussed, but body can form up to 16 carbon normally. After 16 carbon, we use one more mechanism to introduce double bond. Is clear? We use fatty acyl CoA desaturate. We use right now. Normally, body can form up to carbon 16 palmitic tak. After that, there is different mechanism to further elongate the chain. What will you use? Anybody who wants to add up or has studied earlier in class 1, 2 or something else, tell me. He will be honored with one free chapter. Yes, yes. सुनती नहीं बात क्या करवाता हूँ अभी तो पहले ये खत्म कर ले एक बार फॉर फर्दर elongation what do you need beyond 16 carbon you need endoplasmic reticulum and microsomes to further elongate the chain, right? How will you elongate? The enzymes are elongases. Elongases. They further elongate the chain. That is why the name is elongases. And in this, you add successive malonyl-CoA to the chain, right? So what will you finally get? What will you finally get? Even more long chain fatty acid, right? And that is okay now. Now we'll do with the regulation of fatty acid synthesis. Regulation. We have only 15 minutes left. Regulation. How will you regulate it? Regulation is done at different levels, right? One is at enzyme level, the fatty acid synthesis regulation. How will you regulate it? How will the body know? What happened? You are laughing, Kunishka. What happened? Any questions? Yeah. So, how will you regulate it? How will the body know? I have to synthesize fat or break down fat. For synthesis, you need fatty acid synthesis complex. For breakdown, you need beta-oxidation. How will the body decide, or the intelligence of the body decide whether I will go for the synthesis or breakdown? It happens at multiple levels. One is enzymatic level. Second is at end product level. Right? Third is at dietary level. Right? Anything left? Dietary level, enzymatic level, end product level, hormones and metabolites. Hormones and metabolites. These all will regulate the fatty acid synthesis, right? Metabolites, let's start from this, from reverse manner. Metabolites are, one is acetyl CoA, second is NADPH, third is ATP, right? These are the three raw materials. When you don't have enough NADPH in the body, will the body go for fatty acid synthesis? Answer is no. If these three raw materials are sufficient enough, then will the body will go for fatty acid synthesis. Otherwise, body will not go for fatty acid synthesis. Right? Right? You have to build this building. You need bricks and cement. If bricks and cement are not there, how will you synthesize this building? Simple thing is that, right? Then enzymes and hormones. Tell me the enzymes that regulate this. Major enzyme is first one enzyme, that is our acetyl CoA carboxylase. This was the first enzyme, na? Hope you remember, right? How will you regulate at this level? First thing is that, tell me, anybody, how will you regulate at this level? Sandhya Rani आई है आज बैठी है, yeah. How will you regulate at this level? Acetyl CoA carboxylase. Tell me. This is a committed step, right? And this occurs in two forms. One is inactive form, and the second is active form. Right? We are discussing the enzymatic regulation of fatty acid synthesis, right? And we are doing it. The first enzyme regulator is acetyl CoA carboxylase. It exists in two forms. One is inactive and other is active. This active form favors polymerization, and this inactive forms favors depolymerization. Whenever the substrates are in the body that favor the polymerization will go for synthesis. But when the substrates are favoring the inactivation of this enzyme, the body will not go for fatty acid synthesis, but it will go for breakdown via beta-oxidation, right? Tell me the things that favor this and tell the things that doesn't favor it. Tell me. Tell me. This is favored by citrate. Citrate favors the polymerization and favors the fatty acid synthesis. And the products that favor depolymerization are palmitoyl-CoA, and one is palmitoyl-CoA and second is malonyl-CoA. So just to recall, first is enzymatic regulation at the enzyme level, right? Enzyme is acetyl CoA carboxylase, right? This enzyme exists in two forms. One is active form and other is inactive form, right? Active form favors the polymerization and favors the regulation of synthesis, but inactive form depolymerization and it doesn't favor the synthesis. And the products that activate it are citrate. So citrate will go favor the fatty acid synthesis, and the second two are palmitoyl-CoA, and they will favor the inactivation of this enzyme, right? If we have this palmitoyl-CoA, where it is coming at the end product, we have excess fatty acids, right? This is 16 carbon, right? The end product is there. If we have sufficient amount of fatty acids, why will the body go for the fat synthesis? So this end product will finally regulate via inactivating this enzyme, and the body will not go for fatty acid synthesis, right? And enzymatic regulation is done, metabolites done, because NADPH is sufficient, the body will go for it. Acetyl CoA is sufficient, the body will go for it. ATP is sufficient, the body will go for it, right? Enzymatic now, hormonal. Tell me the hormones that favor synthesis and which don't favor synthesis. Anybody? सुना है जरा मीठा खाएगा मोटा हो जाएगा, बोला जाता है. लड़की जरा मीठा मत खा मोटी हो जाएगी, शादी नहीं होगी तेरी, बोला जाता है. क्यों बोला जाता है? Tell me. Tell me. There are. Tell me. Any guesses? Any guesses? Hormonal regulation. Again, this enzyme acetyl CoA carboxylase. It has two forms. One is active and other is inactive. Right? And this active form, you can say it is A form, and this is not, you can say inactive form. Right? Active form is D-phosphorylated form. Means it doesn't contain phosphate. And the inactive form is phosphorylated form, right? And this dephosphorylation is favored by insulin. and this phosphorylation is favored by glucagon, epinephrine, norepinephrine, right? ज़्यादा मीठा खाएगी तो क्या होगा? More sweets. If you eat more sweets, what will happen? The body will secrete insulin, and if more insulin is there, it will favor dephosphorylation, right? Dephosphorylation will what will do? It will activate this enzyme and it will form fatty acids, right? In the starting of the previous class, we discussed if you are taking excess glucose or excess carbohydrates, and if you are taking excess proteins, they are diverted for fatty acid synthesis, right? Clear? So we discussed the enzymatic regulation. Now we discuss the hormonal regulation. Metabolites are done now at dietary level. End product we discuss. End product is palmitic 16 carbon. When it is sufficient, it will inhibit the cycle from the front. That is end product inhibition. Now dietary level. What will you eat that the body goes for fat synthesis? What will you not eat? The body starts breaking the fat. What will you eat? Tell me. Tell me. Any gases? Any guesses? We have already discussed keto diet. Hope you remember in beta-oxidation and other steps. Tell me dietary manipulation of the one diet is high carbohydrate and low fat. One diet is this. You are eating more carbs and low fat, and one is high fat diet. Tell me which will favor fatty acid synthesis and which will favor fatty acid breakdown. Takkar, any guesses? Sarwar Adil, any guesses? Tell me. Yes. Sandhya Rani, any guesses? How will your body move? This favors the synthesis of acetyl CoA carboxylase. High carbohydrate, right, and low fat will favor acetyl CoA carboxylase. What will it do? Acetyl CoA carboxylase. It will convert acetyl CoA into malonyl-CoA, and malonyl-CoA will enter the fatty acid synthetase complex. And what will it do? It will start forming the fat. So if you are eating more carbs and low fat, the body will go for fatty acid synthesis, right? And if you are eating high fat diet or keto diet, just for say 60% or 70% fats, 20% protein and 10% carbs, right? The body will inactivate this enzyme. Inactivation will, what will it will cause? It will cause fatty acid breakdown or starvation. Starvation is also there, I forgot. If you are fasting, the same mechanism will happen. This will be, acetyl CoA will be inactive. When it will be inactive, the fatty synthesis will not happen in the body. Clear? So this is all done.